So we start off today. Good evening. So we start off today with the origin of life as the next chapter in cell biology. The origin of life is the process by which living organisms develop from inanimate matter, which is generally thought to have occurred on Earth between 3,500 and 4,000 million years ago. Methane, which has helped develop life on Earth between 3,500 and to 4,000 million years ago, occurs in Jupiter, Saturn, and interstellar space. Origin of life is known as biobiosis. Means biobiosis is the development of living matter from complex organic molecules that are themselves non-living but are self-replicating. First life evolved 3,800 to 4,200 million years back. There are several theories about the origin of life. These are theory of special creations, theory of eternity, theory of catastrophism, cosmozoic theory, or theory of panspermia, theory of spontaneous generations, and upper and Haldane theory. The theory of special creation was proposed by Hebrew et al. And supported by Father Cervers, the Spanish priest. According to this theory, life was created by supernatural power, either once or at successive intervals. The theory of eternity of life, also called the steady-state theory, states that a life has ever been in existence as at present and will continue to be so forever, changing only in one form. It neither had a beginning nor will have an end. Steady-state theory does not accept the paleontological evidence that presence or absence of a fossil indicates the origin or extinction of the species, represented in quotes as an example the case of coelacanth latimeria. According to the theory, scientists like Richter, Preyer, Helmholtz, Arrhenius, Hoyle, and Bonte believe that life is immortal. Sudden creations of life from inorganic material was supported by George Cuvier. This theory is called theory of catastrophism or cataclysm. According to the theory, the abrupt faunal changes geologists saw in rock strata were the result of periodic devastations that wiped out all or most extinct species, each successive period being repopulated with new kinds of animals and plants. Cosmozoic theory was proposed by Richter 1865 and supported by Arrhenius 1908. Cosmozoic theory is also called theory of panspermia and spore theory. This theory states that life had reached the Earth from some other heavenly body in the form of resistant spores, or simple organisms called cosmozoa, in meteorites or in spaceships. Theory of spontaneous generations or abiogenesis or autogenesis states that life originated from non-living things in a spontaneous manner. For example, insects were believed to originate from dew, frog and toads from moist soil under the influence of sun, butterflies from cheese, and fly and maggots from flesh. The theory of abiogenesis was believed and supported by Thales, Anaximander, Newton, Descartes, and Van Helmont. Theory of spontaneous generation was disproved by Francisco Redi in 1668, Avi Lazzaro Spallanzani in 1767, and Louis Pasteur in 1867. Francisco Redi, 1626 to 1698, performed a series of experiments to disprove the theory. He replaced meat of fish, eel onto clean muslin coverings and demonstrated that while flies laid eggs in muslin, maggots and larvae appeared only when those eggs were transferred to the meat and allowed to hatch. He concluded that maggots developed or develops only from pre-existing flies and were not spontaneously generated by any other form of material. In 1765, Abbé Spallanzani, an Italian scholar, prepared flasks of meat broth, which were boiled for several hours and then sealed. The broth remained clear for months, and when the seals were broken and the broth tested, it was shown to be free of microbes. Louis Pasteur in 1864 used a swan neck flask and prepared a meat broth in this flask and boiled it for several hours. He then left the flask unsealed on a laboratory bench. The flask was not sealed and there was a free exchange of air with the environment, so the system did not lack oxygen. Still, the swan necks remain free of microbial contamination for months because their swan necks were shaped so to trap viable microbial particles and to allow only air to enter the flask. After several months, when you broke the neck of one of these flasks, contamination by air and proliferation of microorganisms in the fluid ensued. This experiment thus disproved the concept of spontaneous generation completely. Pasteur is known for germ theory of disease. The process of destroying all living organisms is called sterilization. Sterilization is done to surgical instruments to kill all living organisms and to remove all pathogens present in the instrument. All three scientists, Red-Eye, Spallanzani, and Pasteur, developed theory of biogenesis. Right? All the three scientists developed theories of biogenesis. According to theory of biogenesis, life originated from pre-existing life. The modern hypothesis of origin of life was formulated by Haeckel. This idea was elaborated in the chemical theory by two workers. Independently, that was done independently. A Russian biochemist, AIIMS, in 1923, and an English biologist, J.B.S. Haldane, in 1928. It was summarized by Apparatus in its book, The Origin of Life, published as an English edition in 1938. Apparatus and Haldane state that spontaneous generation of life under the present environmental conditions is not possible. Earth's surface and atmosphere during the first billion years of existence were radically different from today's conditions. In the initial atmosphere, Earth was produced. First life arose from a collection of chemicals through a progressive series of chemical reactions. Solar radiation, heat radiated by Earth, and lightning provided energy for evolution of molecules. Modern theory of origin of life was propounded by Apparatus, which is based upon chemical evolution. The Apparatus-Haldane theory, also called protobiogenesis, was experimentally supported by Stanley Miller in 1953. Modern views regarding the origin of life included origin of Earth and its primitive atmosphere, chemical evolution, chemogeny, and biological evolution. It is considered that the Earth was formed about 4,600 years ago, 4,600 million years ago, for basic requirements of the life to start to arise our primitive atmosphere with little or no oxygen, O2. Some notable milestones in origin of life, approximately origins in millions of years. The origin of the universe was about time in million years as 10,000 to 20,000 through Big Bang. Bemida, 931 of Cosmic Era, a solar system about 4,600 million years ago, Earth 4,600 million years ago, about an origin of anaerobic life about 4,200 million years ago, and anaerobic photosynthesis bacteria is 3,500 to 3,800 million years ago. Beginning of traces of oxygen was about 3,800 million years ago. Oxygen producing photosynthetic cyanobacteria was 3,300 to 3,500 million years ago. Eukaryotes with a nucleus 1,600 to 1,200 million years ago. First land plants were 459 million years ago. First mammals 220 million years ago. Man and Homo sapiens. Homo sapiens was about 3.5 million years ago, and traces of oxygen 3.8 million years ago. Right, and now to the next page. Right chemicals, including water, various inorganic ions and organic molecules, and energy source at infinite time. The energy needed for chemical evolution on the primitive Earth mainly came from solar radiation and cosmic rays, electric discharges, volcanic eruptions, heat, etc. Early Earth had innumerable free atoms of all those elements which are essential for formation of protoplasm. The lightest atoms of nitrogen, hydrogen, oxygen, etc. formed the primitive atmosphere. Free atoms combined to form molecules and simple inorganic compounds. Primitive atmosphere was producing atmosphere because hydrogen atoms, most numerous and most reactive, combined with all available oxygen atoms to form water and live in no free oxygen. Water and ammonia were probably the first compound molecules of primitive Earth. Chemical evolution, also called chemogeny, involves the synthesis of simple organic molecules. With slight lowering of the surface temperature of the Earth, the lighter elements interacted to form water, methane, first organic compound, ammonia, CO2, HCN, etc. The early compounds interacted and produced simple organic compounds such as simple sugars, nitrogen bases, amino acids, glycerol, etc. Simple organic compounds were formed with the help of external sources, UV rays, cosmic rays, lightning, etc. acting on the mixture. Experimental evidence for formation of a simple organic compound was given by Stanley Miller. The apparent-Haldane theory suggests that complex organic molecules would have been formed through a series of chemical reactions in the Earth's primordial soup. Synthesis of complex organic compounds from their simple constituent molecules were experimentally proved by Stanley Miller with the help of stimulating experiments. The apparatus used by Miller is called a spark discharge apparatus. Miller passed an electric discharge and a mixture of methane, ammonia, hydrogen, a ratio 2:1:2, and water, steam. By simulation experiments, Miller synthesized about 15 amino acids and identified as glycine, alanine, glutamic acid, and aspartic acid. The energy used in the Miller-Urey experiment was the electric spark. The first simplest organic compounds to arise were methane, methylene, acetylene, etc. Hydrocarbons carried to the Earth's surface by rainwater, the simple organic compounds then would have accumulated in the ocean as a warm, dilute soup. The organic water-rich mixture of organic compounds was termed by J.B.S. Haldane 1920 as hot dilute soup of organic substances or prebiotic soup. The largest organic molecules, which were synthesized abiotically on primitive Earth, later came together and formed large colloidal aggregates. Proteinoids are protein-like substances formed by polymerization of amino acids under inorganic conditions such as heating to over 140 degrees centigrade. The formation of protein molecules can be considered a landmark at the origin of life. Colloidal aggregates were named microspheres by Sidney Fox and KORAH-BIOTECH by Operin. The model proposed by S. Fox of proteinoid microsphere for protocells is widely accepted because of its following significances. Such proteinoid microspheres arise from monomers rather than from polymers obtained from organisms already in the air, biota. As is true for the usual experiments with the KORAH-BIOTECH droplets. 2. This model suggests that proteinoids are informational and it shows the origin of communication, which may be intercellular or intergenerational communication. Nucleic acid, formed by the polymerization of nucleotides, are the basis of life as these represents the genetic material of an organism and is the molecular basis of heredity. Nucleoproteins gave most probably the first sign of life. Biological evolution or biogeny. Biogeny involves formation of prokaryotes or eukaryotes. More complex coarse weights and microspheres function as precells or prebionts, the first living form, named protocell or cobiont, a protobiont, originated in this primitive ocean. Amino acids, sugars, glycerol, and fatty acids gave rise to polymers, which may have assembled into spherical structures called protobionts. Alright, and now on to free atoms. Those are hydrogen, carbon, nitrogen, inorganic molecules, hydrogen, water, methane, ammonia, carbon dioxide. Simple organic molecules or precursors will be sugars, fatty acids, glycerol, amino acids, nitrogenous bases, purines, pyrimidines. Complex organic molecules of biological origin, a sugar plus sugar given polysaccharides, fats, which are from fatty acids and glycerol, amino acids giving proteins, nitrogen bases, sugars and phosphates giving nucleotides, adenosine phosphates, nucleotides giving nucleic acids, right? And then coacervates, operin, bioperin, aggregates of large complex organic molecules capable of growth and divisions, eubions, nucleic acid controlled coacervates. Free gene will be self-replicating nuclear protein complex. First primitive cells, lipid proteins, membrane bound units with enzyme controlled metabolism and nucleic acid regulations without organized nucleus. Leads to prokaryotes, chemoheterotrophs, and anaerobic due to absence of free oxygen from the environment. Eukaryotic cells with well-organized nucleus, multicellular organisms. Flowchart. Main steps in the origin of life according to Oparin, Haldane theory, and modern views. Protocells were prokaryotic unicells which had naked DNA, protein manufacturing machinery, mode of energy liberation, and its utilization. Protobionts made of polypeptides are called microspheres, those of lipids are called liposomes, and those of combination of polypeptides, nucleic acids, and polysaccharides are called coacervates. The original prokaryotes were probably from protocell. The first prokaryotes were anaerobes and chemoheterotrophs. Chemoautotrophs were formed when environment over the earth became cooler. The organisms performing chemosynthesis are called chemoautotrophs. And simple one-cell organisms, somewhat similar to today's cyanobacteria, were present on Earth about 3,600 million years ago. Atmosphere became richer in oxygen due to photoautotrophs. Free-living eukaryotic cell-like organisms originated in the ancient ocean, presumably about 1.5 billion years ago. Primitive eukaryotes led to the evolution of protists, plants, and animals. The oxygen present in the modern atmosphere must have been liberated as a result of photosynthesis of green plants. The most important condition for the origin of life is the presence of water, because life can originate from abiogenetic materials in water. There is no life on the moon because there is no water. Life originated in the ocean water, presumably about 3.7 billion years ago in pre-Cambrian era. The prokaryotes evolved before the eukaryotes. The oldest known fossil cells are about the same size as modern prokaryotes. Some of the oldest known fossil cells appear as parts of stromatolites, which are formed today from sediments and photosynthetic prokaryotes. The earliest autotrophs must have been anaerobic chemoautotrophs. The first organisms to give off oxygen were probably cyanobacteria. When a primitive bacterium is exposed to oxygen gas, it dies. Organisms that can grow with or without oxygen gas are called facultative anaerobes. Organisms that are poisoned by oxygen gas are obligate anaerobes. Organisms that cannot grow without oxygen gas are obligate aerobes. Prokaryotes were limited in genetic variability because they reproduced by binary fission. Haeckel, 1894, proposed to create a separate subkingdom Protista to include all unicellular eukaryotic plants and animals. Prokaryotes are more diverse than eukaryotes in terms of metabolism. Of the planets of our solar system, only Mars is supposed to have life, but no evidence of life has yet been found by the scientists. And that will be the end of this chapter. Read well, understand well, prepare your notes, and get back to me just in case you have any doubts. Place your doubts in the comments section, okay? Best wishes.